Hi everyone, myself Dr. Kirti here. Welcome to Pathology channel. Today we are going to learn about the most important topic in general pathology about apoptosis. Okay. So before that, if you want to see many lectures of mine and also to understand the key concepts, please try to subscribe my channel and also get notified. So coming to the topic apoptosis, it's a most important question for both undergraduate and also in the postgraduate level. So to understand the concept, it is very essential to recollect in your exam period. Okay, here in this class, I'll make it very simple. What are the concepts of pathology and what are the pathways in apoptosis, and then how can you approach and write this particular question during your exam. Okay, so coming to the topic apoptosis, actually speaking, that by the definition, it is a type of a programmed cell death. Okay, so it is in our body. What happened? The apoptosis will happen like a natural process. Okay, it is a natural process where the cell commits suicide by itself. It's a suicidal program. Okay, suicidal itself by the controlled and very clean manner, unlike the necrosis where there will be a messy environment. So in necrosis, what will happen? The cellular contents will leak out outside the cell, right? Then it will cause inflammation. But whereas in apoptosis, the cellular content does not leak out. So it will form like apoptotic blebs and it will be phagocytized. So that's why there will be no inflammation will be seen in the apoptosis. So it clean by itself where it does not harms its neighbor cell. Okay, that is the most important point which is differentiating the apoptosis and necrosis. So a little bit about the history. In 1972, the John Kerr and et al. Have discovered the word that is apoptosis. That's a programmed cell death. By the Greek word means that is a falling off of the leaf. Why they call it? Because they notice there are some cells in our body where it dies itself by its own but does not cause any damage to the surrounding neighbors. Okay, and it happens in a very peaceful cell death. That's why they call it as a Greek name of falling off means apoptosis. Okay. So next, moving on to the key features of apoptosis. So if you take features of apoptosis, there are 4 key features. Okay. The first one is activation of intrinsic enzyme. So once there is any particular signals which is coming from outside the cell, it will activate your intrinsic enzymes within the cell. Once that is activated, it will degrade your cell. Okay. So this enzyme will start to degrade your cell. And finally, it will form apoptotic bodies. What are apoptotic bodies? These are nothing but the cytoplasmic projection where it will have nuclear contents, DNA contents and everything. So that is known as a formation of apoptotic bodies. So one cell will divide into multiple parts. Okay. So then finally there will be phagocytosis. Phagocytosis means it will be happened by the macrophages where it will engulf your apoptotic bodies and digest it. That is the phagocytosis. So these 4 are the key essential features for apoptosis. What are those? Activation of intrinsic enzyme, degradation of the cells, formation of apoptotic bodies and phagocytosis. So we'll see in detail about each in the concepts. Okay. So next, moving on to the causes of apoptosis. So they are divided into 2. One is physiological, the other one is pathological. So why the apoptosis should be a physiological, why it should be pathological? Because there are certain processes in our body where it will keeps our cell turnover very correctly. So mediated by apoptosis. If the apoptosis exceeds or if it is lacking, then it will cause certain disease. So that we are naming it as pathological causes. So the first one is physiological. So we'll see about the embryogenesis. So what will happen in embryogenesis? So here during the process of development in the embryo, for example, if you take the Mullerian duct, okay, so they are the progenitor of uterus. So they will be deleted in males. Okay, Mullerian duct will be deleted in males, whereas Wolffian duct will be deleted in females. So that is the best example they are giving for embryogenesis. And also your digital webs. So it will be fused together, and then there will be apoptosis and it will give you 5 digits and everything. And also like a heart where it will form like first like a solid organ, then there will be a division of your right atrium, right ventricle, left atrium, left ventricle, and also your GA. Okay, GA system. So all this process happens by apoptosis. by the natural phenomenon happening during your embryogenesis. So the second one is hormone dependent. So what is hormone dependent? So here, if you take an example, like during our menstrual cycle, the layer in our uterus will try to apoptosis and will be shedding off. So that is also one example. And also ovarian follicle atresia. And the next one is like lactational hyperplasia in women, where the breast and also the uterine changes will be seen. And the next one is cell population. If you take hematopoietic elements, there should be particular number of cells. So if it exceeds, then it will be going into certain disease. So to maintain that, the apoptosis will happen. And the other one is like removing the harmful agents. So that is also there. And the next one is immune response. So what is the immune response? For controlling your autoimmune diseases, cytotoxic T cells, T lymphocytes, and everything will be managed by apoptosis. And finally, the senescent. Okay, the aged cell will also undergo apoptosis so that it can maintain the turnover. Next is gametogenesis. That will happen like spermatogenesis, and all those things will come under gametogenesis. Okay, so these are the physiological causes of apoptosis, which will maintain our body in a normal mechanism. So next, moving on to the pathological process. So what are these? The elimination of parenchymal cells. So that will be atrophy is the best example. And then the elimination of a damaged DNA by your P53, and the elimination of a misfolded protein. There will be example like amyloidosis, Alzheimer's disease, Huntington disease, and Parkinson's disease. So these are trinucleotide and also the misfolding disease. So this should be removed by the process of apoptosis. If that is not happening, then it will lead to the particular disease. Okay. So and also in killing the viral cells like HIV, HBV, HCV, HTLV, all these things will also, the apoptosis will happen. So if there is a failure, it can lead to certain infection. And there will be elimination of neoplastic cells and also in transplant rejections. So these are the examples of a pathological apoptosis. Okay. So next, coming to the morphology. So here in the morphology, what are the difference you can see? The electron microscopy, there will be a cell shrinkage. If you take a necrosis, there will be everything enlarged, because I told you there will be a sodium pump failure and water content will come inside. So there will be a enlargement. So here what will happen? The cell shrinkage will happen. And finally, there will be a condensation of your fragmentation of nuclear chromatin. So once the cell is shrinkage, okay, so then it will lead to condensation of your nuclear, and nuclear fragmentation will happen. And finally, it will form the cytoplasmic blebs. So these cytoplasmic blebs are the one where it will contain both your cytoplasmic organelle and also the nuclear organelle and DNA. So all this will form the cytoplasmic blebs. So once that is formed, it is called as apoptotic bodies. So this apoptotic bodies will be engulfed by your macrophages, that is known as phagocytosis. So this is the same key features which we saw in apoptosis. Okay. So next, moving on to the light microscopy, what will be the changes? There will be a round to oval mass and eosinophilic cytome will be there. And then pycnosis, there will be shrinkage of nucleus. So like a dot, your nucleus will turn off. That is known as pycnosis. And there will be no inflammatory reaction, remember, because the cellular contents are not leaked here. So they formed into apoptotic bodies and they are engulfed by the macrophages. So that's why finally there will be phagocytosis. Okay, so if you see in this diagram, you can see here there is a normal cell which has all the nucleus and endoplasmic reticulum, mitochondria, and everything is there. So that will undergo, first there will be a pycnosis and nuclear condensation of the chromatin. And what will happen? It will form few membranous blebs. Your plasma membrane will form a blebs. So then it will fragment into many things. So that is known as apoptotic bodies, which will have both their cytoplasmic and nuclear organelles. And finally, these are detected by your macrophages and phagocytosis will happen. Finally, it prevents your inflammation. So these are the steps in apoptosis. Okay, so next, moving on to the signaling pathway. So we have to know there are n number of pathways. The most important is extrinsic, intrinsic pathway. Okay, the others are inflammatory or infectious process, perforin or granzyme pathway, P53 activated apoptosis, endoplasmic reticulum. So actually speaking, the apoptosis is regulated by many biochemical pathway. One thing is, the most important is death signal, and the other one is pro-survival. So if there is any imbalance in this, the pro-survival things are heightened up and the death signals are more voluntary, that will lead to your apoptosis. Okay, so there should be a balance always to be maintained between your anti-apoptotic and pro-apoptotic signal. Okay, so next coming to the role of mitochondria in apoptosis. Why it is very important? Because all the intrinsic pathway happens within the mitochondrial thing. So it is very important to know before entering into intrinsic pathway. You have to know about what is the role of mitochondria in apoptosis. So they are speaking about something cytochrome C. Okay, so what is the cytochrome C? They are messenger cell death. Okay, so they are the messenger of cell death. So they are like a power plant. If you imagine, they are like a power plant where the normal protein of cytochrome C will helps to make the energy within the mitochondria. So if there is some particular stress coming and acting within the cell, what will happen? This will act like a alarm. Okay, this will act like a fire alarm. So it will call other proteins for the help. So come here, like that. It will call, which is the thing that is cytochrome C. So the cytochrome C will be the normal thing which is present in your plasma membrane inside the plasma membrane. So once there is a damage, they will act like an alarm and call other proteins for the help, and they say it is a switch here, it's a time to die the cell. Okay, and let's do it in a very clean way. That is your apoptosis. So you understood what is the cytochrome C? It is the messenger of cell death. Once it is activated, it will finally lead to cell death. Okay, so they are present in the membrane of the plasma membrane. So once it comes out, it will induce your apoptosis. Okay, so before that you have to know what is BCL2 and BH3 family proteins. So BCL2 family proteins are BCL2, BCLXL and MCL, and BH3 only proteins are BAX, BIM, BIT and BCD. So here in this diagram you can see the viable cell and apoptosis. This is the role of mitochondria and cytochrome C in your intrinsic pathway. That's how we are going to see. Okay, so in this side you can see the viable cell will have a survival signal. So once this growth factor comes and binds to its receptor in the membrane, it will lead to the production of anti-apoptotic protein, that is your BCL2. It is an anti-apoptotic. So what it will do, the cytochrome C are present within inside the cell membrane, right, plasma membrane. So there will be no leakage of cytochrome C and no induction of your cell death. So once there is a irradiation, there is a damage to the plasma membrane, any DNA damage or something is happening, so it will lead to activation of your pro-apoptotic, that is your BH3 only sensor protein, and it will antagonize your BCL2. So BCL2 is an anti-apoptotic, so that will go down and your pro-apoptotic will come up. So this will lead to, there will be an activation of your BAX and BAC channel, where the cytochrome C from inside the plasma membrane will come outside. So once this come outside, I told you it will act like an alarm. So there will be a leakage of cytochrome C and it will call the other proteins for help, and finally there will be activation of caspases and there will be cell death, that is your apoptosis. So you understood the concept? Cytochrome C is very important for mitochondrial role. So I told you it is present within the cell membrane. So inside the cell membrane, once there is a DNA damage or something, the cytochrome C will be activated. How? By those signals which will promote your BAX, BH3 only sensor protein, and it will antagonize your anti-apoptotic, that is your BCL2 protein. So when that is happening, it will activate your BAX and BAC, and there will be a channels will be created within the plasma membrane that will leak your cytochrome C from inside the cell to outside the membrane. So once that is come outside, it will act like an alarm and it will induce your apoptosis. Okay, this is the concept. So again, the same as a flow chart. Once there is a DNA damage, there will be inactivation of your BCL2 anti-apoptotic protein, and there will be a shift of your cytochrome C from inside to outside the cell membrane. There will be activation of a BAX pro-apoptotic factors, and activation of caspase will happen, and finally apoptosis. Understood? So coming to the phases of apoptosis, we saw already there is a initiation phase and execution phase. In initiation phase only we are having your intrinsic and extrinsic pathway, and finally this intrinsic pathway and extrinsic pathway will combine and form your execution pathway. Okay, so these are the phases of apoptosis. So what are the phases? There is an initiation phase, there is an execution phase. In initiation phase you have intrinsic pathway and extrinsic pathway, and finally this intrinsic pathway and extrinsic pathway will combine to go to execution phase and lead to apoptosis. So first we are going to see about intrinsic pathway. So here the intrinsic pathway are mainly controlled by your anti-apoptotic protein, that is your BCL2 family. So what is the trigger which is responsible for intrinsic? That is your deprivation or withdrawal of the growth factor or survival signals. And the second one is DNA damage by radiation, cytotoxic anti-cancer drugs, and also hypoxia. And the third one is accumulation of excessive amount of misfolded protein. I told you, like all the Huntington's disease, Parkinson's, Alzheimer's, etc. And increased intracellular free calcium level. So these all acts like a signal to the mitochondrial pathway. So you can see in this diagram there is a plasma cell membrane. So once there is any intrinsic stimuli that is coming and activating your BH3 only protein. What are the BH3 only protein? They are the pro-apoptotic. So that will antagonize your anti-apoptotic factor like BCL2 and all those things, and thus it will activate your backs and back, and finally it will create a channels within your membranes. That from the channel there will be a leakage of the cytochrome C and other proteins from the inside the cell to outside, and that will form the apoptosome with the complex of APAF, that is your apoptosis activating factor 1 oligomerization along with the SMAC. Okay, so this will activate your caspase 9. Once the caspase 9 is activated from procaspase to caspase, it will lead to the other pathways. Okay, so this is the main concept. So BCL2 is the one which will regulate your mitochondrial permeability. Okay, once the BCL2 is out of control, it will cause the channels within the mitochondria. So that's what they're trying to say. So there should be a proportionate balance between your anti-apoptotic genes and pro-apoptotic genes. Once the pro-apoptotic genes raised up, then it will lead to the apoptotic pathway. So first there will be a mitochondrial damage by BH3 only protein. Once there is a mitochondrial damage, it will activate your pro-apoptotic signals, backs and back, and finally thus they block the anti-apoptotic protein and they will create the channels within the mitochondrial membrane, and that will leaks out the cytochrome C and other proteins from inside to outside the cell, and thus the cytochrome C will binds with the APAF1 and forms the apoptosome multimer complex. So once that complex is formed, it will activate your caspase 9, and thus it initiates the caspase to the intrinsic pathway, and it activates also caspase 3, and finally it will go to the execution phase. So caspase 3 is very important. Caspase 9 and caspase 3 are coming in your mitochondrial or intrinsic pathway. Remember only that caspase 9 and caspase 3. Okay, so next moving on to the extrinsic pathway. So we saw about intrinsic pathway which is wholly happening within the mitochondria. So extrinsic pathway are initiated by the extracellular signal. Okay, they are activated by extracellular signal. So here if you take many cell, okay, in our body which will express the depth receptor on their surface. So there will be a depth ligand and depth receptor. Okay, so once the ligand is like a key and whereas the receptor is like a lock. So once the ligand and receptors are locked in, it will open the door for cell death. That is a concept. Okay, the ligand and your receptor will combine and it will open the door for cell death. That is your apoptosis by the FAD. We will see that one step by step. So here that will trigger your apoptosis. Okay, so these receptors will contain a death domain. What are those receptors? That is TNFR1, FAS, that is your CD95, and FAS-L is a ligand where CD95 ligand. So this ligand will be expressed on all the cytotoxic T cells. So T cells will express these ligands. Okay, So what is the main function of the extrinsic pathway? They are against the self-reactive lymphocytes and you're killing the target cells like virus and tumor cells. So here in this diagram you can see there is a ligand FASL and there is a receptor FAS. So once those both are combined, it will activate your FAD, that is your caspase 8 and everything will come set. So once the FAD is activated, it's the death domain. Okay. So once that is activated, it will convert your procaspase to activate into caspase. So in the mitochondrial pathway, we saw caspase 9 and caspase 3 are the most important thing. So here we are seeing as caspase 8. So once the procaspase is converted into caspase 8, then it will executionate all the other caspases and finally leads to apoptosis. Okay. This is all about your extrinsic pathway. So what are the steps? First there will be, I told you, the key and the lock should bind. So that is your ligand and the receptor should bind. So thus it will activate your door to the cell death, that will activate your caspases. What are the caspases? 8 and 10. In mitochondrial pathway we saw about 9 and 3, whereas in extrinsic pathway we are seeing about 8 and 10. So once the inactive form is converted into active form of 8 and 10 caspases, it will downstream all the caspases in the execution pathway. So there is something called as FASL that will bind to the FASS receptor in the cytoplasmic T cells. So finally there will be activation of FAD. What is FAD? FAS associated death domain. So I told you death domain will be open for the cell death. Okay. So that will activate your caspase 8 and finally it will combine to your execution pathway. So in intrinsic pathway we saw caspase 9 and caspase 3. In extrinsic pathway we saw about caspase 8 and caspase 10. So all this will go to your execution phase. So next we are going to see about execution phase. So what will happen in execution phase? The activated caspase 8 will come and it will activate multiple number of the downstream caspases and finally that will lead to the cell death. Okay. So next we have to also know about what are the inhibition of extrinsic pathway. That is your flip and flies. That is like a FAD like interleukin 1 beta converting enzyme. So these are the inhibitory protein for your extrinsic pathway. So they actually inhibit by binding to the procaspase 8. So it will be not converted into your activation forms. Okay. So next execution phase of apoptosis. I told you again, intrinsic pathway will happen, extrinsic pathway will happen. What are the main caspases you should know in both, and finally it will go to the execution pathway. So caspase 9 and 3 are the one which will happening in, which is most important in intrinsic pathway, and caspase 8 and 10 are the most important in extrinsic pathway. So they both will combine to come into the execution phase. In execution phase, they activate the procaspase of 367 to active form of caspase 367. Okay. So what are the roles of 367? They will divide it into three fragments. Okay. So there are three functions of it. What are those? That they will activate your endonucleases and nuclear proteins like laminin, and they break down your cytoskeleton proteins. By that it will damage your nuclear fragmentation. So once these are activated, 367, it will lead to nuclear fragmentation and also form the apoptotic blebs. I told you there will be a bubble kind of thing will come where it will contain both cytoplasm and the nuclear fragments, that is called your apoptotic bodies, and also there will be a karyolysis, like a nuclear fragmentation will happen, and finally it will lead to apoptosis. So apoptotic blebs will be formed and thus it will be engulfed by your phagocytosis. So this is the process of apoptosis. So here is a Robbins diagram where you can see mitochondrial pathway summarized. Mitochondrial pathway is there and your extrinsic pathway is there. So in mitochondrial pathway, any stimulus, that is your cell injury like DNA damage and anything, that will go and downregulate your BCL2 family and it will upregulate your back, that is your pro-apoptotic signals, and thus it will create a cytoplasmic membrane channels will be created and it will shift the cytochrome C within the cell to the outside the cell. Once the cytochrome C comes out, it will act like an alarm and it will call all other proteins for the help to activate the cell death. So once this has happened, then it will activate your procaspase 9 and 3 and it will convert it into activation form. And if we see in the extrinsic pathway, what will happen? All the signals like a lock and key. I told you receptor and ligand will combine. Once the receptor and ligand are combined, it will initiate all your caspases and your executional caspases will come, and finally it will go to execution pathway where it will activate your other caspases like 3, 6, 7, and it will lead to your endonucleosis activation and breakdown of your cytoskeleton, and finally thus the cytoskeleton breakage and your nuclear fragments will combine and form the apoptotic body that will be like a cytoplasmic bleb, and that will come as a separate fragment, that is your apoptotic body, where it will contain your cytoplasmic organelle and also your nuclear material, and those apoptotic bodies will be engulfed by the phagocytes, that is your macrophage. So this is the concept of apoptosis. So how will you diagnose the apoptosis? Mainly by the electrophoresis, okay, that where it will have a step ladder pattern. Okay, that is the first thing. And the tunnel, what is tunnel? Terminal deoxynucleotide transferase biotin D UTP NIC and labeling. So this is also one of the other investigation. And also you can detect by HNE, Feligin and also acridin orange. So these are the investigation for diagnosis of apoptosis. What are the main enzymes? Cytochrome C, activated caspases, annexin 5 and propidium iodine. Okay. So next, coming to the disorder associated with apoptosis, we already saw what are the physiological causes and pathological causes of apoptosis. So from that you take an idea and make it very clear. So if there is a decreased apoptosis, it will lead to the cancer like follicular lymphoma in your lymph node and also autoimmune diseases. Okay, if there is a defect in apoptosis. So if there is an increased apoptosis, what will happen? It will cause certain neurodegenerative disease like your Alzheimer's, Huntington's and Parkinson's. And if it is happening in the ischemic injury, it will lead to your myocardial infarction and CVA. If there is a death of viral infected like HIV is an other example. So next, moving on to the difference between apoptosis and necrosis. So we already saw in the last class about necrosis. So here we discussed about apoptosis. So we'll see what are the difference between these two. The cause is mainly often physiological in apoptosis and pathological in necrosis. So biochemical events, there will be a DNA fragmentation by endonucleases, and here there will be a impairment of cessation of iron homeostasis, and lysosomes will be intact in apoptosis where it will be leaked in necrosis. And morphology, it will be single or small clusters in apoptosis where it involves a groups of cells in necrosis. And the cell size, there will be a shrinkage and fragmentation, condensation of nucleus, apoptotic blebs and phagocytosis in apoptosis, whereas in necrosis there will be enlargement, cellular swelling will happen, and finally it will lead to lysis. Integrity of cell membrane will be maintained in apoptosis where it is not maintained in necrosis. And the nucleus, there will be a fragmentation of the nucleus will happen in apoptosis, whereas here I told you three features: pycnosis, karyorrhexis and karyolysis can happen. And cellular content may be intact in apoptosis, but it will be enzymatically digested in necrosis. And next, the fate of dead cells is ingested, whereas it is also ingested. And the inflammatory response will be no in apoptosis, where it will be present in your necrosis. And DNA electrophoresis will show the step ladder pattern in apoptosis, and there will be smearing pattern in necrosis. And the tunnel will be positive in apoptosis, where it will be negative in necrosis. So what are the other programmed cell death which happen in our body? Like apoptosis, necroptosis, pyroptosis, onychosis, netosis, entosis, pyronecrosis, autophagy associated cell death, and ferroptosis. So we'll see about each in further classes. So in this class we discussed whole about apoptosis, and in previous class you saw about necrosis. So here is a summary. Whenever the apoptosis is asked in your exam, you have to write about the definition first, then you have to write about what are the physiological and pathological causes, and then moving on to the morphology, what are the features will be seen in H&E and electron microscopically, and the next one is your signaling patterns, pathway you have to write intrinsic, extrinsic, all those pathway, and then what is the role of mitochondria in apoptosis, and finally you will move on to the steps of intrinsic pathway and extrinsic pathway, and then your execution pathway. Next, what is phagocytosis, and at last about the diagnosis and diseases of apoptosis. So this should be the order of writing in your exam of when the question is asked about apoptosis. I hope so you guys understand the concepts of apoptosis, and we'll see in the next topic with some other new. Okay, until then, thank you.